Presenting, secreting, and sensing the same molecule allows cells to achieve versatile social behaviors. It's wonder in biological world that how different types of cells use the common and minor collection of circuit elements to achieve a range of diverse functions. Here, let's see some examples. Bacteria sense their group or assembly by secreting and sensing an autoinducer. So autoinducers are signaling molecules that are produced in response to changes in cell population density. As the density of quorum-sensing bacterial cells increases, so does the concentration of the autoinducer. Another example can be human pancreatic beta cells. Human pancreatic beta cells secrete and sense insulin. Lymphocytes or T cells can be another example. They induce a monoclonal immune response by secreting and sensing cytokine interleukin 2, which is known as IL-2, to regulate their growth. So in these cells, there are circuits that simultaneously secrete and sense same molecules, and these type of circuits can commonly be found across species. So these cells use the same core signaling circuit pattern, which is known as the secrete and sense circuit. In research, it has been found in some cases the cells secrete and sense same molecule when communication is needed within itself, which is known as self-communication, but not with the neighbors. Whereas in other cases, it showed the reverse condition. Cells secreting and sensing molecules to communicate with neighbors, but not for self-communication. Again, some cases also showed cells communicating both within itself and also with the neighbors simultaneously. The question is, if the cell needs self-communication, then what's the reason behind secreting molecule? Why doesn't the cell work entirely within intracellular environment? To get answers of these questions, this study was done to learn more about the sense and secret circuit motif. Getting a clear idea about this pattern can help us to understand various range of cellular and biological function. If we can get deeper understanding of the versatility of this intracellular circuit, then we may explain its recurrence across many species in the nature. For this study, yeast was considered as a simple cell model. A synthetic secrete and sense circuit pattern had been constructed in budding yeast. The circuit enabled the yeast to secrete and sense the mating pheromone. The variable key factors of the circuit were altered technically throughout the experiment. The key factors were the rate of secretion, receptor quantity, positive feedback relationship between sensing and secreting, and signal degradation. The key factors were regulated to know how they facilitate the communication within a cell versus its neighbors. It was a single cell measurement, but with two assessment approach. Number one, self-communication, measuring the extent to which a secrete and sense cell responded to its own secreted signal, and number two, neighbor communication. That means how the secreted cell responded to the signal secreted by its neighbors, and these two measurements were compared to understand the cellular behavior. The results showed that the secrete and sense circuit motif work in both social and asocial ways. Cells which showed low secretion rate and low expression level of low affinity receptor, they acted that way to prohibit self-communication. For example, in quorum sensing, we can see this kind of example. So these cells are considered as purely social. In case of purely asocial behavior, the epithelial cells self-communicate through signaling loop called autocrine signaling loop. They express large amount of epidermal growth factor EGF. From this diagram, it can be seen the cells that had the highest secretion rate with the highest number of receptor, they are considered as asocial. The result from the positive feedback shows that the relationship between the sensing and the secretion is stable in two behavioral ways. So the cells present in the population can be one of the two groups, to be in an inactive state or be maximally active. When the signal degradation was being incorporated, it enabled the bimodal activation, which means the cell population divided into two different activation states. At this intracellular circuit element, the secrete and sense circuit mechanism not only reveals about the self and neighbor communication, but also it shows how it can take turn to various population level behaviors. So it shows how the fundamental cell signaling circuits can enable multicellular behavior in a population. Quorum sensing have been used to demonstrate cellular behaviors and engineering goals, for example, cellular logic gates. So building synthetic signaling circuits, which represents natural signaling circuits, can provide us more information about how interaction of molecules are linked to cells and then higher level macroscopic multicellular behaviors. Thanks for watching.